Good day, this is the Professopher, CBMD PhD, and today's Monday, December 26, 2022. It is 3:08 and 47 seconds, a.m. Eastern Standard Time, and today's research, maternal microbiome. So let's hop to it here. The evidence of horizontal gene transfer between human maternal microbiome and infant gut microbiome. Date December 22nd, 2022. Source: Cell Press. Researchers have discovered a new mode of vertical mother to infant microbiome transmission, where microbes in the maternal gut share genes with microbes in the infant gut during perinatal period, starting immediately before birth and extending throughout the first few weeks after birth. As a horizontal gene transfer allowed maternal microbiome strains to influence the functional capacity of the infant microbiome in the absence of persistent transmission of the microbial strains themselves. As this is the first study to describe the transfer of mobile genetic elements between maternal and infant microbiomes, the study also, for the first time, integrated gut microbiome and metabolomic profiles from both mothers and infants, and discovered links between gut metabolites, bacteria, breast milk, substrates. As this investigation represents a unique perspective into the co-development of infant gut microbiomes and metabolomes under the influence of known maternal and dietary factors. The gut bacteria promote the maturation of the immune system and part through the production of microbial metabolites. The development of the infant gut microbiome follows predictable patterns, starting with transmission of microbes from the mother at birth. In addition to immune system maturation, microbial metabolites also influence early cognitive development. The perinatal period represents a critical window for cognitive and immune system development promoted by maternal and infant gut microbiota and their metabolites. Nevertheless, the co-development of microbiomes and metabolomes during the perinatal period and the determinants of this process are not well understood. And to address this knowledge gap, the research team tracked the co-development of microbiomes and metabolomes from late pregnancy to 1 year of age using longitudinal multiomics data from a cohort of 70 mother-infant dyads. They discovered large-scale mother-to-infant interspecies transfer of mobile genetic elements, frequently involving genes associated with diet-related adaptations. Infant gut metabolomes were less diverse than maternal metabolomes, but featured hundreds of unique metabolites and micrometabolite associations not detected in mothers. Metabolomes and serum cytokine signatures of infants who received regular but not extensively hydrolyzed formula were distinct from those of exclusively breastfed infants. The infant gut harbored thousands of unique metabolites, many of which were likely modified from breast milk substrates by gut bacteria. Many of these metabolites likely impact immune system and cognitive development. The pregnancy was associated with an increase in steroid compounds, including gonatal hormone derivatives and intermediates of bile acid biosynthesis, several of which were independently linked to impaired glucose tolerance. Although infant gut metabolomes were less diverse than maternal metabolomes, the researchers detected more than 2,500 infant unique metabolomic features. Moreover, they identified numerous infant-specific associations of bacterial species and fecal metabolites, including neurotransmitters and immune modulators. Researchers were surprised to find that the maternal gut bacteria that were rarely observed in infants contributed to the infant gut microbiome structure. Researchers also found evidence that prophages, dormant bacterial phages, or viruses that reside on bacterial genomes contribute to the exchange of mobile genetic elements between maternal and infant microbiomes. The authors say that the maternal microbiome may shape the infant gut microbiome through horizontal gene transfer, apart from classical vertical transmission of strains and species. Moreover, the identification of distinctive metabolomic profiles and micrometabolite interactions in the infant gut constitutes a platform for further study of microbial contributions to infant development. The research study limitation was that the researchers did not consider changes in diet and lifestyle between pregnancy and the postpartum period, which may have affected microbiome and metabolome alterations. In future studies, they plan to further explore linkages between bacteria and metabolites and investigate strain-specific bacterial metabolic output using isolated bacteria in vitro. As taken together, the integrative analysis expands the concept of vertical transmission of the gut microbiome and provides new insights into the development of maternal and infant microbiomes and metabolomes during the late pregnancy and early life. And this is the Professopher, Professor of Microbiology, Bioinformatics, and Pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.